Hello lovelies, in this video we're going to be going through your AQA Separate Science Foundation Biology Paper 2, the paper that we've written for 2024. Now if you haven't had a go at this paper already, what we want you to do is to pause after each question comes up, have a go at it yourself on a bit of paper, play the video and then watch us work through the answers, and anything you don't get right you can put onto flashcards.
The diagram below shows the brain. And it wants us to label A, B and C on the diagram above. So it wants us to use words from the box below. So just making sure that if we can, maybe we could eliminate some words from the box that we don't need, that we know are wrong answers to start with. So the pituitary gland is not labelled. I know that that's there. I've just circled it in purple, so it's not the pituitary gland. The hypothalamus is also not labelled, it's just above the pituitary gland in that space there. So that means I know that those two have not got a label, so it can't be one of those two. So I've cut it down to 3. So now it's just about knowing the answers and which one goes in which. Being able to label the brain is just something we've got to know. So A is the cerebral hemisphere. The part that's kind of in the middle of the brainstem that then joins onto the spinal cord is the medulla, and then part C is the cerebellum. Drinking excessive amounts of alcohol can lead to memory loss. Name the part of the brain that is affected by excessive alcohol consumption. Okay, so it's given us a clue in the question stem that large amounts of alcohol lead to memory loss. So this first part is really just a backwards way of asking you which part of the brain is associated with memory. So again, this is just something we have to know, and hopefully we know the functions of these. So the part that's associated with memory is the cerebral hemisphere. The cerebellum is to do with kind of like your automatic functions like breathing, and the hypothalamus is to do with controlling things like homeostasis, like temperature and stuff. So the cerebral hemisphere is the part that's associated with memory. Give 2 reasons why it's hard to treat someone suffering with memory loss or other brain damage. So why is it difficult to get access to or trying to treat the brain? So when we obviously treat things, normally we have scans and tests, and then we have the ability to be able to try different drugs and see if they work, or we can do surgery to try and fix things and stitch things and repair things or replace things. But obviously with the brain, all of this is so much harder because it's hard to see, it's hard to access. You don't want to have to like cut someone's head open if you really don't have to, or you don't know what you're looking for or where you're going, where you need to access it. It's hard to rely on the scans. We only have like MRI scans to try and see what's happening inside the brain. It's really hard to interpret those. They're not always really clear. Nervous tissue is quite hard to repair. Obviously, it's special. It has this ability to conduct the electrical signals. It's one of the only tissues in the body that can do that. So it's quite difficult to replace or repair that as a tissue. And we still have a limited understanding of how the brain actually functions and how it works. We know, we're learning all the time, but actually in terms of really understanding exactly what goes on where in the brain, knowing it for sure, is still really hard. So we don't know exactly what we're dealing with at all times. So any of these are kind of marks in this, and you'll see with this there's quite a lot of options you can say because it's quite a kind of a big concept to understand and try to explain, and there's lots of different parts that make it difficult. So any of the 2 things in the mark scheme, as long as you've explained that, what the difficulty is or why it's hard, or what can we not do, or what is it difficult for us to do, and why that's an effect on the fact that it's hard to treat the brain or brain damage. Any of those 2 will get you marks. So I'm going to go with it's difficult to repair nervous tissue and our understanding of brain function is limited. The eyes send information about light to the brain. Name the type of cell that carries impulses from the eyes to the brain. Okay, so now we've kind of switched back to being more about the nervous system, and it's asking us what type of cell carries impulses that send the information about light from the eyes to the brain. And they've missed out a word here because they just said impulses, but it's basically what cell carries electrical impulses. Hopefully we should know the optic nerve is what connects the eyes to the brain and sends those electrical impulses and information along. And so we can say it is a nerve cell. Or you can use the word neuron, that's also fine. The two diagrams below show how light is focused in people with different eye defects. So we need to name the eye defect in each person. So we're supposed to hopefully be able to recognise from the diagram what's wrong with each of these people in terms of their eye defect and work it out. So the defects are to do with focusing light. So it's nothing to do with any part of the eye other than the lens and how they're not able to focus the light properties. So we're talking short-sightedness and long-sightedness here. So in person B, the light's being focused too far from the retina, so far away past the retina. And in person A, it's not far enough, or it's too short. So too long, the distance is too long in person B, and too short in person A, and that's how I remember whether it's long-sighted or short-sighted. So too short, it doesn't quite meet the retina, that means that person is short-sighted, and too long past the retina, that means that person is long-sighted. This question is about plant growth. Plants will grow towards the light. Give the name of this response. So any response where someone's going towards something is going to be positive. And this time it's saying they're going towards the light. So hopefully we know that photo would be the prefix for this because that's always about the light. And then it is a tropism because that is what we refer to as kind of growth responses in plants are all tropisms. So this is positive phototropism. If you didn't get the positive part, that's fine. It doesn't necessarily need that part for the marks. The most important bit is the phototropism part. Explain how bending towards the light helps plants to grow. So we need a reason why, and we need to link the idea of why going towards light might help growth. So we should hopefully know that when plants get more light, they can carry out more photosynthesis. And for growth, that's what they need to do because they need to produce the sugars that they get from photosynthesis in order to produce molecules that can help them grow cells, to provide the energy they need to grow cells. So more light equals more photosynthesis. So if they're bending towards it, more light's going to be hitting on and absorbing by all of those cells, and therefore they can do more photosynthesis and produce more sugars, and that's how they grow. So anything about when you're talking about a question that links light and plant growth, it's just a question about photosynthesis. That's what it wants you to say. So we're going to say obviously bending towards the light helps them to get more light for photosynthesis. The student wanted to investigate the effect of light had on new germinated seedlings. They set up 3 Petri dishes of newly germinated mustard seeds on a wet cotton wool pads in 3 different conditions. Dish A was in normal conditions with light all around during the daytime. Dish B was in total darkness, and dish C was in partial light with a light source only coming from the right-hand side. So we kind of have normal conditions and then we have no light for dish B, and dish C has got light just coming in from one direction only, from the right-hand side. The diagrams below represent the growth of the seedlings after 5 days. Label each dish with the letter A, B or C that matches the conditions they were grown in. So because it's got normal conditions and light all around, dish A is going to have normal shoot growth. We know that dish C, because it's going to have one direction only, it's going to bend towards the light. The previous questions we had kind of led us to this idea that we're going to be talking about the plant responses and hormones and the bending towards the light source. So if it's going to bend towards the light, think about how it's coming from the right-hand side. The only one that's bending towards the right-hand side is C. And I put the arrows on to show where the light was coming from. If it was coming from the right-hand side, for, like we said, for treatment C, so that must be dish C. So then we've got to choose between the other two. We've got kind of normal thickness, same as C, and then we've got tall and skinny in the other one, which is the difference. So although you might just go naturally one looks normal and one looks different, so one will be the normal conditions and one will be darkness, just think about why that bottom one would be the dark group. And that's because if there's no light, plant shoots will grow up fast. They'll grow up fast and really tall and skinny quickly to try and look for light because they think they're still underground. So that makes sense that that's why they look like that in B. And so therefore that bottom diagram must be the ones that were grown without light. And then A is just the normal. So that's the top picture. Give 2 conditions that need to be controlled in this experiment. So light is obviously the thing that was being changed. So we can't talk about light because being controlled means it needs to be kept the same. So what else other than light could affect the growth of plants that we would need to keep the same? So think about the things that could affect photosynthesis or could affect the growth of the plant. So light, which we also know is about the thing that was changed, so we can't say that. Temperature, water, and carbon dioxide concentration. Carbon dioxide concentration obviously affects the rate of photosynthesis, but because they were just grown in normal conditions, other than they weren't grown kind of sealed in anything, then there's no way you could control that anyway. So it is going to be temperature, because we know that could speed up the rate of growth potentially, and water. So we know that they were put in Petri dishes with wet cotton wool. So volume of water in each dish would also have been really important. Name the piece of equipment and the units the student should have used to measure the growth of the seedlings. So they're measuring an increase in growth over time, so an increase in shoot length, so like how tall, how long the shoots were. So if you're measuring the length of something, you have to use a ruler, so that's easy for the first bit, which is the equipment. Then we have to think about the units. So small, and there's going to be small changes. So these seedlings are just starting to grow. They're only small, they're only short, and there's going to be small changes day to day in the length. They're not going to grow meters in length. So we should be saying millimeters, and we should know that millimeters is going to be a small enough measurement that we can see the small changes in growth that will happen in the day to day. Select the time interval the student should have measured the seedlings and give a reason for your answer. So it's really asking how long in between repeated measurements should we have left it, or should the students have left it, and then the reason why. So weeks would be too long. There would have been loads of growth happening between weeks that you would have missed. But hours is the opposite, it would have been too short. This is a bit like Goldilocks here. Not too hot, not too cold, just right in the middle. So too long would have been weeks, too short would have been hours. You wouldn't have seen a change in growth from hour to hour. But days, there would be changes within days that you could be measurable, and it wouldn't be too long and there wouldn't be too much growth happening. So that's our reasoning. So because there'd be too much growth in a week or in weeks, and there'd be no noticeable change within hours. Explain how auxins help to control the growth of the seedlings. So this is asking us about, do you understand how auxin works? How does the hormone control growth? So what happens is light coming from one direction is going to cause auxin to diffuse to the shaded side, wherever the opposite to where the light is. And then that auxin there causes growth, causes elongation and growth of those cells on that side, whereas on the other side they don't, where the light is, and so it causes the shoot to bend forwards towards the light. So this is how auxins help to control the growth of plants. They'll move to where the shaded side is, and therefore that causes uneven growth. So make sure they grow towards the light if the light is only coming from one direction. Obviously in the ones where there was no light, there's no auxins controlling the growth, other than that it's evenly distributed, so it will be all shaded, so they'll grow straight up. Same for if there's the light coming from all around, it's evenly distributed, so they'll grow straight up. So auxin moves to the shaded side of the shoot. It stimulates cell growth, so the shoot bends towards the light. So we get 1 mark for talking about auxin movement to the shaded side, and we get 1 mark talking about how they actually work, so they cause that cell growth. The diagram shows the evolutionary tree of part of the big cat family, Felidae. Wants us to state which big cat is most closely related to the jaguar. On the family trees like this, the most closely related ancestor will share a branch or be on the closest branch to the one you're being asked about. So in this case, it shares a branch with the lion, so you can see they've got a join there, so that's our answer. The image below shows a picture of a clouded leopard. Now it wants us to state the genus of the clouded leopard. The genus is always the first part of the binomial name because we use the genus and species taxonomy groups to make the binomial name. So Neophelis is the first part of the binomial name for the clouded leopard. And then lastly, state the species name of the snow leopard. So the species name is the binomial name. So we need both parts of the name. We don't just say the second part. It doesn't make any sense without the genus. That's why we have the binomial name. It has to come in 2 bits. So if we're doing the snow leopard's binomial name or species name, it's Panthera uncea. Common mistakes made with these questions is being careful when copying. You are copying down Latin names, so be careful with the spelling, but also make sure you use capital letters and small letters where you should as well. And remembering that the species name is the binomial name, so you need both parts. The snow leopard is quite different from the other cats. It lives in very cold, snowy, mountainous habitats. Suggest a feature of the snow leopard that helps it to survive in its environment. So this is an adaptations question. That's what it's really asking here, about features that help an organism to survive. So it's also a suggest question. So we need to use our knowledge about adaptations and also the question information which tells us it's very cold, snowy, mountain habitat. So we need to give specific adaptations to this information about it being very cold. So adaptations we'd expect to know or have learned for cold environments would be something like a fat layer to insulate, thick layer of fur or hair, or having a small surface area to volume ratio to try and reduce heat loss. So I'm going to go with a thick coat of fur to reduce heat loss. We need to make sure we add how it helps it survive. So it's only 1 mark, but you can't, I'd rather than just put a thick coat of fur, making sure you've added that extra bit so you're linking it directly to why it's an adaptation to the cold and how it helps it to survive. Unlike all other cat species in the same genus, the snow leopard cannot roar. There is a debate about whether it should have its classification changed. So this would mean it would not be in the same genus anymore as the others. Describe a method scientists could use to determine if the snow leopard is significantly different enough from the other cats to be put in a different genus. So when it asks to describe a method, it's talking about what are the steps in the process and how would we be able to tell or decide if something is significantly different in terms of relatedness from other organisms. So normally we compare and talk about similarities in their DNA. That's how we make these phylogenetic trees. And how do we look for differences or similarities? We compare the base sequences. So we need to sequence the DNA of the cats in the genus, so all the cat species in the genus, and the snow leopard as well. And then we would need to compare the base sequence of their DNA to see how different they were. Variation may arise due to the geographic isolation of a species. Geographic isolation of members of one species can lead to a new species evolving. Name this process. So this one is just asking us, what is it called when a new species is formed? So the word evolving is already in the question, so it can't be evolution. The answer is speciation, and the clue is in because it has the word species almost in the name. But evolution and natural selection is the process by which evolution happens. And evolution doesn't always necessarily mean that a new species will evolve. It just means a change in a species over time. But if it happens to 2 different populations and they change so much that they can no longer interbreed with each other, then we get the species, because our species definition is a group of similar organisms that can breed to produce fertile offspring. So if they can't breed to produce fertile offspring anymore, then they will be a different species. So only then do we get speciation, and it normally happens when the populations are separated, like what it's talking about with geographic isolation at the start of the question. So they have to be separate for a time, let natural selection and evolution happen over time in separate populations that can't breed together, and then eventually they'll be changed so much over time that even if they're put back together, they wouldn't be able to breed to produce fertile offspring anymore, and so we've created this new species. Explain how geographic isolation of members of one species can lead to a new species evolving. So now it's asking us to explain, like what I was just saying, how geographic isolation, so how the separation of those members of one species and then natural selection and evolution can then lead to that new species evolving. So we need to link this idea of isolation, and then how a new species forms from it. So the process is speciation, and we've got this idea of like there's two populations of a species, A and B. They're one species, but they got separated geographically for some reason. And so either side of this, in these two new environments, we're going to have different environmental conditions. And then natural selection is going to take place in either side of the new environments. And if we're forming a new species, then it means they must not be able to breed with each other anymore. So that is enough really for 3 marks. This isn't a really long answer question. We're not having to go through the whole process of like evolution. We're just giving an overview for 3 marks. Then we're going to say that the different areas are going to have different environmental conditions. We're going to briefly describe what natural selection occurs so that only the best adapted members in each environment survive and breed. And then what makes them new species is eventually the two populations can no longer interbreed, so they are new species now. Now, I've probably said more than enough for 3 marks, but this is still a decent level of detail and kind of what you need to be talking about if you're talking about speciation. Students were asked to estimate the number of buttercups in a sports field that is 50 metres wide and 90 metres in length. They used a 0.5 metre by 0.5 metre quadrat like the one shown in the image below and placed it in an area of the field that contained some buttercups. One buttercup flower has been circled for you. Identify the number of buttercups that the student should record in this quadrat. Tick one box only. Okay, so we've got the circled one, and then we've just got to count the rest of the number of buttercups we can see inside the quadrat, and then we've got to pick which one of those numbers is right. So we've got our 1, the one that's circled, then 2, 3, 4, 5, 6, 7, and then 8 is there down the bottom. So the best and sort of easiest way to do this, and what you should have done this when you've done this practical before, is kind of go along each row of the quadrat, and then you don't miss any. Make sure you go along each row. Make sure you can count every single time you see a buttercup. 3 I've crossed out don't count because they're outside of the quadrat. So our answer then is 8. Use the quadrat to calculate an estimate for the total number of buttercups in the field. Okay, so to do that, first of all, we need to calculate the area of the field. So the area of a rectangle is obviously just length times width, so 50 times 90. So that gives us 4,500 metres squared. Then we need the area of the quadrat. So that's 0.5 by 0.5 metres, so that gives us 0.25 metres squared. So to do the final sum, we need the 8, which is the number of buttercups in this quadrat, divided by the area, so number of buttercups per this area that we counted them in, which was 0.25 metres squared. And then we're going to times that up by the whole area of the whole field. So that gives us 144,000 as an estimate of the number of buttercups in total on the field. Describe a more accurate method the student should use to estimate the number of buttercups in the field. So the clue here comes from the part of the question where it said they placed the quadrat in an area of the field that contained some buttercups. So they found some buttercups and then put the quadrat down. So that's biased, and you know that that's not the way that you should do this practical because it needs to be random. So it wants us to describe, so give steps in a process, give the detail, a more accurate method, so one that would be random, for example, and way they should use to estimate the number of buttercups in the field. As well as being random, the placement of the quadrat, they only did 1. So us using that 1 quadrat was not acceptable. You have to repeat it, do it quite a few times to get a representative sample of the area, and then you would calculate the mean number of buttercups in all of those different spots that you put down in the field, and then you do the same calculation we've just done. So for 3 marks, we're going to have to describe how we're going to do it randomly, how we're going to do many of them, and then that we're going to calculate a mean from that. So my first mark is going to come from saying use a random number generator to produce random coordinates in the field. Now, there is just a mark in the mark scheme here for it being or mentioning that you should do it randomly. So however you've said the word random, as long as you've got it in there and you've made it clear that you would do something in your method to make your placement of the quadrat random. You'd place at least 10 quadrats. So this is about there being a large number, so that's the mark in the mark scheme. And then we would calculate a mean in order to then across those quadrats, and then we would estimate our number. So always in these practicals, when they ask about improvements, how can it be made better, more accurate, you need to be thinking about large number of quadrats. Did they do that? Yes or no? Did they do it randomly? Yes or no? And you can always say repeat and calculate a mean, okay? Because that will always get you a mark here, because the more you do that in any experiment, that's going to make your results more reliable, okay? So when we're thinking about this practical in particular, these are the questions and these are the things they always want you to say. Buttercups have a shiny surface to their petals which reflects UV rays into the centre of the flower. This increases the temperature of the flower. It has been shown that bees prefer to visit warmer flowers as it helps them to maintain their body temperature. Explain how the shiny surface of buttercups is an adaptation. So remember, an adaptation is a feature of an organism that helps to increase its survival or reproduction, because then it's passing on its genes to the next generation. It wants reasons why that shiny surface is going to be an adaptation. And obviously it gives us information in the part of the question that says that the bees prefer to visit warmer flowers. And it's the shiny surface that helps to increase the temperature of the flower. So if your flower is warmer, more bees are going to come and visit, which means you get more pollinators. And if you get more pollinators coming to the flower, then that increases your chance of sexual reproduction. It increases the pollination that's going to happen between carrying your pollen to other flowers as well. So the first mark's going to come from saying that we understand that it increases the temperature, which attracts more bees. Don't get any marks just for talking about that it increases the temperature, because it tells you that in the question. So you've got to link the increase in temperature to more bees coming and visiting the flower. And then our second mark is coming from how we're saying this is an adaptation. So remember, how is it helping it to survive or reproduce? So pollination by bees increases chance of successful sexual reproduction. Even if you just said sexual reproduction, or it increases the chance of pollination, that's fine. The students noticed that there seemed to be fewer buttercups around the edges of the school field where there were lots of trees and hedges. Suggest a reason for this. So this is a suggest question. They're often the hardest ones on the paper. You're going to need to bring in your own ideas and information given to you in the question and put it together to provide an answer. It's not necessarily going to be something, just a piece of content or knowledge that you can just regurgitate. So it said there's lots of trees and hedges around the edges of the school field, and there were fewer buttercups there. So we need to link this idea of why having lots of trees and hedges might reduce the number of buttercups near them. So there's a couple of options here, and this is quite common with suggest questions. There's not normally only one right answer. So we could mention that the idea that these trees and hedges are going to shade that area of the grass, so there's less light for photosynthesis. Or it could be due to competition, so bigger, larger plants nearby, there'll be less nutrients available and less water available in the soil, and therefore the growth of the buttercups would be worse. So because this is an explain, so we need a reason, we need to make sure we do say not just that it's shaded. For 2 marks, we need to say shaded or less light, and that means less photosynthesis, so they're not going to grow as well. Or the competition, we say obviously less nutrients or water, and therefore they won't grow as well because of that. So I picked the light answer. So the trees and hedges create shade, so there is less light available for photosynthesis. The local council want to try and reduce the number of weeds on the sports field. Identify the hormone they should use and give a reason for your answer. So this is a long way of saying what hormone can be used as a weed killer, because they're using it to kill the weeds. And we need to give a reason. So we need to explain why that hormone particular that we've chosen, why can it be used as a weed killer? So what does it do to the weeds? So let's go through these options. Ethene is used for ripening fruit. Gibberellins is used for like seed germination. So logically the only one left is auxins, and they can be used as weed killers because they are dealing with plant growth. So auxins work as weed killers because they cause the plants to grow too fast. So that's our reason. They cause the plants to grow too fast, and so they die. Can't just talk about causing plant growth, have to have this idea that it grows too quickly or too fast, So they grow up too tall and they can't support themselves and they fall over basically, or they grow too quickly and use up all the energy reserves and then they can't function. So the idea of too fast, too quickly is what you need to get the mark here. Beta thalassemia is a recessive inherited disorder which reduces the production of hemoglobin in the body. The family pedigree shows the inheritance of beta thalassemia in one family. So my first thought is, what is a recessive inherited disorder? It's the one that needs two alleles to be the same to be present in the phenotype. So it's the one where you have to have two lowercase letters, for example, in order to have the disease. And if we're thinking about maybe a question we might get asked later, we might not, but I'm thinking, okay, if it reduces the production of hemoglobin, that means there'd be less oxygen being transported if we don't have as much hemoglobin in the body. So we've got the parents in the first generation, then we've got their children and their grandchildren in the third generation. Tick the row that correctly identifies the sex and genotype of person B. So we need to go and find person B on the family tree. So they are a filled in circle, which means that means from the key they are a female with beta thalassemia. So it's just looking at sex determination here. So the sex chromosomes, we're not talking about the alleles for beta thalassemia at this point. So we know they've got to be female, and so it's got to be one of those two rows. But we've got either XX or XY. So this is really just a question about, do you know the sex chromosomes for male and female? So XX is the sex chromosomes female and XY is male. So we're ticking that bottom row. Explain why sperm determine the sex of offspring at fertilization. So the reason why is what we need to try and say here. Why does sperm chromosome determine if an embryo has XX or XY? And this is simply because sperm can either contain an X or a Y, depending on which chromosome is present out of the two options. Eggs can only contain X chromosomes because female cells can only donate an X chromosome to each gamete. So if you have a Y chromosome present in the sperm, then the fertilization will happen and you'll get XY and therefore a male offspring. If you have an X in the sperm, then you'll get XX and you'll have a female offspring. So this is basically what we need to say. We need to say that sperm can carry a Y or an X, and having a Y chromosome determines male, and if you don't, then you're female. The gene responsible for beta thalassemia can be represented by the letter B, capital B. Capital B represents the dominant allele and little b represents the recessive allele. Both individuals F and G in generation 2 are heterozygous for beta thalassemia, so they have big B, little b. Use the diagram and your knowledge to explain why. So we need to be able to explain why they must both be big B, little b or heterozygous, and what evidence can we get from the family tree. So if you find F and G, we can see that they do not have the disease, but they have had an affected child. So this is very common with recessive disorders. In questions like this, they'll get you to go and find two parents that do not have the disease and they've had a child that does have the disease. So that must mean they both have to be heterozygous because they both must have a copy of the lower case allele, the recessive allele, in order to give both of them to give a copy to child I, in order for child I to be little b, or double or homozygous recessive. So we can always start our explanation by saying they are affected, but they produced a child, in this case child I, who is affected. So we can say they must both have had at least one recessive allele in order to both pass on the recessive allele to child I, but obviously they are both unaffected, so they must also have had a dominant copy. Use the Punnett square to calculate the probability that the next child, individuals F and G, could have beta thalassemia. Use capital B to represent the dominant allele and use lowercase b to represent the recessive allele. And they filled in the parental genotypes for us. And then we fill it in as normal. So 2 capital B's, 2 squares that have large b and lowercase b, and then 1 square with 2 lowercase b's. So that then needs to be circled to show that we understand that that genotype would produce an affected child. And then we need to give the probability. So it's 1 out of 4 or 25% chance. that the next child they have will have the homozygous recessive and therefore have the disease. So as you can see in the mark scheme, just doing the Punnett square without circling the little b and giving the information doesn't get you the mark. So make sure you add this every single time you do a Punnett square question. You need to make sure you circle the one thing that the question was asking you to look for the probability for and then label it with the probability. Explain why it's important for people like C or E to have a pedigree analysis in case they decide to have children in the future. So we can see C and E are children of A and B. So their mum has beta thalassemia and so does their brother. It says use percentages or ratios to help illustrate this. So we're going to need to sort of explain what the percentage chance might be of them passing it on depending on what the analysis shows. So why is the reason why they like to have a pedigree analysis? And it's because obviously they might be able to pass on the recessive allele. So they're going to want to know what the probability is of their offspring having the beta thalassemia allele. So if we think about the percentage chance, obviously we've just seen that it could be 25% chance of having a child with beta thalassemia. There could be 50% chance of passing on a recessive allele if you are heterozygous. And if they're homozygous dominant, so they have two capital B alleles, two dominant alleles, then there's 0% chance of them passing on the affected allele to their offspring. So it's all going to depend on what alleles they have and then what alleles their partner has in order to determine what the probability is of having a child with the beta thalassemia allele or having the disease. But only with analysis will they be able to show that, so then they can obviously make those decisions. So an analysis will determine the probability of passing on the recessive beta thalassemia allele. It could be 0% if they're homozygous dominant, or up to 50% if they are heterozygous. A father with the genotype capital B, so homozygous dominant, and a mother with the genotype little b, or homozygous recessive, plan to have several children. Explain why none of their children will have beta thalassemia. So explain why none of their children, the reason why none of their children will be little b. And when it comes to these questions, it's often just easier to draw a Punnett square rather than trying to write it out in words. And often you will get marks for that as long as you annotate it correctly. So let's put those into a quick Punnett square. So we've got two capital B's and two little b's. So it shows that 100% of their offspring are going to be heterozygous. So they're going to all have at least one dominant allele and one recessive allele. So they'll all be carriers, but none of them will be homozygous recessive. So none of them will have the disease. So you can say none of them will have two recessive alleles. You could say they can only get one recessive allele from mum, and so therefore they can't have two because the father can only donate capital or dominant alleles. However you want to explain it, you've got some options in the mark scheme, but if you want to do the Punnett square, that's fine. But you would need to have this extra little bit of text that I've written next to it just to make it clear that you understand that 100% of the offspring are heterozygous, or something like no child will have two recessive alleles to have the disease. Human activities impacting biodiversity. Peat bogs are unique wetland habitats for many species. Peat bogs are being drained for human use. Peat can also be cut into blocks and extracted as seen in the image below. Define a habitat. So this is one of the definitions we just have to know, but a habitat is the non-living part of an ecosystem. So it's the rocks, the soil, the ground, the water. Name 2 biological factors that can affect a population in an ecosystem. So this time biotic means living, so it's a living factor or a living part of an ecosystem that can affect populations. So there's 4 to choose from. Now these are always the same 4, so it doesn't matter about whether it's asking you for one, multiple, it's asking you to explain or list them. The biotic factors that can affect populations, there's 4 that you need to know, 4 main ones that you need to know. So new pathogens, competition for whatever you would like to say, competition for food availability and new predators. Or you can have hunting from humans, which is included in that, so you can't have that as well as predators. But remember, I've got new in front of pathogens and I've got new in front of predators. The idea is that an ecosystem will have evolved along with a certain number of pathogens and the predators that are already there, and there'll be a balance to that ecosystem. If something changes, so a new pathogen arrives where organisms aren't resistant to it, or a new predator arrives which the organisms, the prey species, are not adapted to fight against or camouflage against or hide from or whatever, that can then drastically change the numbers of a population in an ecosystem. But there will be just pathogens that they're resistant to and they've evolved with and that don't affect the numbers greatly. And there'll be predators that they've evolved with that are in the food chain or the food web and that don't affect the numbers greatly, as long as the numbers are in balance. So just making sure we've got that word new in front of those if we are talking about a biotic factor that has a drastic change to the number of the population. Use your knowledge to explain why peat bogs have been destroyed by humans in the last 100 years and why it is important to prevent further peat bogs from being destroyed. So this is an explain question. It wants reasons. There's 6 marks available, so it's one of those long leveled answers. And we've got 2 bullet points to kind of guide us into 2 things or 2 areas that we have to talk about. And we're going to be able to use our own knowledge here as well as get clues from the bit of the question that was at the beginning. So we have looked at peat bogs and peat usage as part of land use in our content for the ecology topic. So you will hopefully know some stuff about peat to start with. But it says peat bogs are being drained for human use. So what are humans using the peat bogs for? Why are they draining them to get rid of the water? What are they using the land for? So it's going to be for things like farming, grazing of animals, building houses or roads and things like that. What are they extracting? What are they cutting it into blocks and extracting it for? Fuel, and as well as that, compost are 2 kind of main uses of peat, cut and extracted like that. So probably some of the easier things to say is, why is it important to prevent further peat bogs from being destroyed? So that's another way of saying, what's the impact on species there? Why is it important that we stop doing this and destroying this habitat? So you're going to get a mark for saying that it is habitat destruction because it says they're a unique habitat for many species. So by draining the water, we're making it not a wetland anymore. We're literally drying it out. So we're destroying the habitat. That's going to reduce biodiversity. That could then potentially affect food chains because you're reducing the number of species that can live in this habitat, which means you might be reducing the number of prey species or a predator species that can then affect the balance of a food chain. And also, a reduction in biodiversity is a bad thing because humans need biodiversity to provide them with resources. So we may think about the fact that there could be unique species there that could have potential as medicines, or we don't want to make sure we don't lose or cause any species to go extinct because they could be useful to us. And then another reason why it's important to prevent this extraction and destruction is because the extraction actually contributes to global warming. So it releases carbon dioxide into the atmosphere, contributes to global warming, which contributes to climate change, and that can have another impact on biodiversity as well. It also says why they've been destroyed by humans in the last 100 years. So yes, this is what they've been destroyed for: farming, grazing, building, fuel, compost. But why? And that's because it's about more people. So remember, when we looked at land use and pollution, all of those things, we talked about kind of why humans are having a greater impact, and it's because the population is increasing, and therefore we need more land and more space to grow crops to feed more people. So that's the reason why they've been destroyed, as well as how they've been destroyed. So reasons why peat's been extracted for fuel and for compost, reasons why it's been drained: farming, grazing, building houses, whichever you want to use, and then the kind of reasons why those have been getting worse, or why more peatland's been used for that, is because of the increase in population. So more people means more land, or more need for fuel, or more need for food. And then the reasons why to stop, we're going to talk about the fact that it's a destruction of habitat, we're going to talk about the impact on biodiversity, and we're going to mention the impact on global warming. So that's my kind of mini plan. This is exactly what I would do if I was in the exam before I wrote it, because it's a lot to write. So here's my answer, and as I read through, I'll highlight where I've kind of put the bits I said I would in my plan. So in the last 100 years, the human population has rapidly increased, and so more land has been used for farming and housing. Peat bogs have been drained so that there is more land to grow more crops and graze more animals to feed the growing population. So this is kind of why it was drained and why the land was used, and I've mentioned the human population. Peat has also been extracted for use as a fuel and as a compost to grow plants. So that is why it's being kind of cut out and extracted. Further destruction of peatlands must be stopped, as it is destroying unique habitat for many species, so it could lead to their extinction. This will reduce biodiversity in the peat bogs, which can disrupt food chains and the ecosystem stability. The extraction of peat also releases carbon dioxide, which contributes to global warming, which can cause a reduction in biodiversity. It is important to maintain biodiversity as humans rely on species for important resources, such as pollination or textiles. You could have talked about medicines or the potential for discovering new medicines in the future or something like that for the last part. But you can see how I've definitely covered both of those bullet points. I've linked ideas together, so this idea of the usage of the peatlands, so for more food and more kind of growing crops or grazing animals and stuff like that. But I've then linked that to the growing population, so why in the last 100 years specifically it's got worse, and it's because our human population has rapidly increased. Then I've linked to this idea of how further destruction of peatlands could keep or continue to reduce biodiversity, and then explain why that's a bad thing. So the destruction of food chains, the releasing of carbon dioxide and the global warming, which even more continues to reduce biodiversity, and then the reason why biodiversity is important, other than just we don't want to cause species to go extinct, but because that can then cause us to not have those important services. Diabetes is a disease where a person is unable to regulate their blood glucose levels. Two people were having their blood glucose. One person has type 1 diabetes and the other is not diabetic. They drank the same concentration and volume of a glucose solution. Their blood glucose was then measured for the next 2 hours. Okay, so we've got a graph with the diabetic person, the non-diabetic person, and the changes in blood glucose concentration over 120 minutes. And it wants us to compare the changes in blood glucose concentration of the two people in the graph. So to compare, we need to compare the similarities and the differences. That means we need to give some similarities and give some differences. And it's asking about the changes in the blood glucose concentration. So how did it change over the pattern over time, and what's different about those patterns, or what's the same? So my first big overall similarity is that both graphs rise and fall within the 2 hours. They both have their peak at the same time at 60 minutes. They both have the same delay at the start, so there's no change for 30 minutes and then it starts to increase at 30 minutes in both. So all of these are similarities. There's enough similarities there, because if there's 4 marks, we need to kind of think about doing 2 of each. So now let's think about differences. So the diabetic person's graph is much higher. So the concentration is higher the whole time and the peak is also much higher. In the non-diabetic person, it returns to the normal levels before or by the time we get to 120, but that does not happen in the diabetic person. And the other final obvious difference is you've got a really steep peak at the start and a much slower descent for the diabetic person, which explains why it doesn't return to normal in the same amount of time. So first I'm going to talk about my similarities. So in both people, the concentration rises and falls over the 2 hours, and the maximum blood glucose concentration for both was seen at 60 minutes after they drank the glucose solution. Then on to my differences. So however, in the person with the diabetes, their blood glucose concentration increased much more rapidly and decreased more slowly than the non-diabetic person. And then finally, I'm just going to add one more difference just to make sure that I've got everything covered, because it's a nice easy one. After 2 hours, the blood glucose concentration of the diabetic patient had not returned to normal, but the non-diabetic patients had. So this was a nice one because obviously there was lots there that we could say, but just make sure with compare questions you've gone back and checked that you've talked about both similarities and differences. And remember, we were talking about changes. So We've used the word more or less or rapidly, more fast or slow. You can use the mark scheme to help you with this, but that's the kind of language we need. Using your knowledge of type 1 diabetes, explain why the graph looks different for the person with diabetes. So this is about using your knowledge. So that means you have to know and be able to explain what type 1 diabetes is. So type 1 diabetes is where they're not able to produce insulin to lower their blood glucose. So it wants reasons why to explain why the graph looks different. And obviously what is different is that the glucose levels are higher and they don't reduce as rapidly as in the non-diabetic person. So how can we explain that? What's not happening using our knowledge of type 1 diabetes? So the glucose is not being reduced as fast or as rapidly, and there is a bigger increase because there is no insulin being produced. So why is the glucose not being reduced as fast? Because it's not being absorbed by the liver because we don't have any insulin. So that's our 2 points. If you just said there's no insulin, that's not going to get you the full set of marks because we need to be able to explain that. We need to say that why is there no insulin? Okay, why does that mean the blood glucose levels doesn't decrease? Because it's not being absorbed by the liver, which is what insulin does. It causes the liver to absorb glucose from the blood. So my first point is because they cannot release insulin into the blood. That will get me 1 mark. So the glucose is not being absorbed into the liver cells. Describe another method that could be used to test to see if a person has high blood glucose levels caused by diabetes. So other than measuring their blood glucose levels, how could you test for diabetes? What are the symptoms of diabetes? Hopefully remember that one of the tests for diabetes is that there is glucose present in your urine when there shouldn't be, and it's only if your body has excess glucose will it get rid of it in urine. So therefore that's why it's a symptom or a sign of diabetes. So we're just going to say testing urine for glucose. Long-term treatment for type 1 diabetes is a pancreas transplant. Long-term effects of type 1 diabetes, and we've got the long-term effects of an organ after an organ transplant. And it wants us to evaluate the option of a pancreas transplant for someone who suffers with type 1 diabetes. So this is an evaluate question, so we need to give pros and cons or advantages and disadvantages, and we then need to sum up with a judgment of our own. So we need to decide what we think is better, the pancreas transplant or not having it. So we're going to evaluate sort of pros and cons or risks in here. So we've obviously got a list of kind of negative effects or negatives or cons of having a transplant. And then all the things in this other column are long-term effects of having type 1 diabetes. So they're the negatives of having diabetes, which we could then flip around to say, turn them into positives of why you would need an organ transplant. What can the organ transplant do to decrease these? And this is the sort of thing that's often common with an evaluate question is not just the information we're being given here, but we need to use our knowledge as well. So we're not just going to be able to basically take this information, just rewrite it out underneath as an answer to the question. It's going to be a leveled answer where we need to give our reasons for and against, link them together, make sure there's logical linking, and then also give our judgment at the end in order to get the top marks. So they all often do this and they give you some information which gets you started, but you need to do something with this information or add your own knowledge together to get it to be a decent answer. So the long-term effects of type 1 diabetes, we can flip those around to positives of having a transplant because you can live longer, you can have more energy levels because they'll have the insulin, so their glucose control will be better. They won't have to have any diet restrictions because they will no longer be injecting insulin. So you don't need to do that anymore if you have a pancreas transplant. All of these are then positive reasons for having the transplant. So the kidney failure and the loss of eyesight doesn't mean that having the transplant will reverse that or take that away. If they've already happened as a result of kind of the effects of long-term diabetes, then you're not going to be able to reverse that. But it shouldn't get any worse. It depends how serious those problems were before a transplant took place. So we have to think about the negatives of a transplant, so the opposite of that. Well, obviously we've got weak immune system and long-term medication here. We need to give more detail because we should hopefully know that the long-term medication is immune suppressants, and why we take them is because obviously there is a risk of rejection. So those are both two kind of negative, sort of bit more detailed negatives of an organ transplant. And then we also need to think of something else, like if you're having a surgery, there's always the risk of infection. So we can always say that as a disadvantage for having a surgery. So then you put these together and we have to try and think of ways we can start logically linking these to form a decent answer and then choose our judgment at the end. So I'm going to start off with my positives of the transplant. So the advantages of a pancreas transplant are that the patient would have increased life expectancy along with a better quality of life, as they would have more energy and no longer need to inject insulin or have diet restrictions. So then I'm going to start saying what some of the negatives might be. So, however, there are risks with the transplant, including infection from surgery and risk of the body rejecting the organ. This can be reduced by taking immunosuppressants, but these must be taken for the rest of their life and can weaken their immune system, which puts them at risk of other infections. So this is where I've linked them together again. So I've said that there's a risk from surgery, there's a risk of rejection, but then I've linked that to the immunosuppressants that they have to take to help with the reducing of the rejection. But then I've also linked that to say that it's a negative because you've got to take it for the rest of your life, and that can be a negative because it can weaken your immune system. And then I've added the extra link there that means that you're at risk from other infections. So you can see how instead of just making that bullet points like it was in the table, so long-term medication, weak immune system, I've turned that into a statement that links those together. So why do you need to take the long-term medication? Why does that create a weak immune system? Why might that be a disadvantage for the person? The patient may also still need treatment for conditions caused by long-term diabetes before the transplant, for example, kidney problems or loss of eyesight. So I've kind of factored in the fact that they don't take those away. They might reduce the chance of them happening, but if they've already happened, then you're still going to need to have treatment for those. So they're my points. I've covered nearly everything we've said about a positive and a negative of both sides, and now I just have to do my judgment. Overall, I think that long-term the transplant is a better option because although there are some risks, the long-term damage to the body will be reduced and the person could live longer and have a better quality of life. So then I've given my judgment, so I've chosen that the transplant is the best option rather than not having it. And then I've given my reasons. And I've said, although there are some risks, which I have said, the long-term damage to the body, so the kidney failure, the loss of eyesight and all the other things would be reduced because it would, and the person could live longer, which they could, and have a better quality of life, which sums up all of those things that we don't have to do anymore if you have a pancreas transplant that you would have had to do if you still had diabetes. So there's lots of different ways you can word your judgment, and it doesn't really matter what you choose. It doesn't matter if you said, I don't think the transplant's a good idea. I think, just treating diabetes with insulin is better. That's fine, as long as you've backed it up with reasons that make sense and are logical. The diagram shows an ocean food chain. So we've got phytoplankton, zooplankton, herring, and yellowfin tuna. And we've got their biomasses underneath. Identify the following from the food chain. A producer. So hopefully we know that a producer is a plant or a photosynthetic organism. It's always something that starts the food chain. So in this case, it's going to be the phytoplankton. Then it wants a predator. So again, definition of a predator is an animal that catches and eats other animals. So the only real options for that here are the herring, which can catch and eat the zooplankton, or the yellowfin tuna, which catch and eat the herring. So clearly we need to pick one of those two, so I'm going to go for the yellowfin tuna. So in case you're not sure, the yellowfin tuna is a fish, obviously, and then the herring is a smaller fish, and then the zooplankton are animal-based, tiny microscopic organisms that are in the plankton, and they eat the phytoplankton, which are the plant, tiny microscopic organisms in the sea. Okay, so some stocks of yellowfin tuna are being overexploited. Explain the impact of reduction in the number of yellowfin tuna on the zooplankton population. So this question is basically saying what will happen to the number of zooplankton if the numbers of tuna decreased, and why? So what would be the reason? So the herring, there would be more herring. So always start from the top and work your way down with these questions. So there would be more herring as there would be less tuna to eat them. So there'd be less of the herring being eaten, so there'd be more of them surviving, so their numbers would increase. That would mean that there would be less zooplankton because there'd be more herring to eat them. So more of them are being eaten because there's more herring. And therefore, Our answer is what will happen to the zooplankton. Their numbers will decrease. Why? Because more herring are going to be eating them, because less herring are being eaten by the tuna. So there's obviously a few ways you can write this. It's quite simply done in the mark scheme. So I'm going to write it as I would explain it. So the number of zooplankton would decrease because then we know we've definitely got that mark for that first point. There'll be more herring to eat them because there are fewer tuna eating the herring. And I've added that last bit because that makes sense to me and that explains how I understand what I'm talking about, but probably wouldn't necessarily need that last sentence to actually get the second mark. Calculate the efficiency of biomass transfer between the herring and the yellowfin tuna. Give your answer to two significant figures. So really, this is asking what percentage of the herring biomass will ultimately end up becoming tuna biomass. So biomass transfer is always going to be the biomass that was actually transferred, or what you get at the end. In this case with the tuna, it's going to be the biomass that they have, divided by how much they took in, the biomass they took in from the herring, which is the 0.13, and then we times that by 100. And that gives us 13.8. And so we have to do it to two significant figures, so we have to round up because 8 will round up to 14%. The data in the graph below shows the annual mass of tuna caught and the biomass of spawning reproductive age of fish in the population since 1950. So we've got two axes here. We've got the total number, or the amount of biomass of fish that are caught, obviously by fishermen, in tonnes, and then we have the mass in tonnes of the spawning age biomass in the population. So the mass of fish within the population that are old enough to reproduce, basically. So just like with the tables and any graphs you should get, always have a look at seeing the patterns that you can spot to start with. So there's clearly been a dramatic increase in the number of total fish that's being caught every year, which is my black dotted line, massively increasing since the 1950s. And there's clearly a decline, or there's fewer reproductive age fish in the population left. So that's been decreasing, whereas the total number of fish being caught has been increasing. So explain how the graph shows that the population of yellowfin tuna is being fished unsustainably. So remembering what being unsustainable means, it's about not leaving enough for future generations. It's about taking more than can be replaced naturally. So we already have our first and second point, the two descriptions of the patterns that we saw in the graph. And then to get our third mark, we have to then specifically say how those two bits of information demonstrate that it's not sustainable. And it's because obviously not enough fish of reproductive age are being left behind to breed to replace the fish that are being caught. So the number of fish being caught each year is increasing, but the number of spawning age fish is decreasing each year. There are not enough fish being left to breed to replace those that are being caught. You've got some more slightly alternative wording in the mark scheme, but that's the idea of kind of having your two bits of description of the data in the graph, the patterns, and then a link to explain how you understand that that means it's unsustainable. State two rules that could be introduced to help conserve the yellowfin tuna stocks. So this really just wants us to give two sustainable fishing methods. It's just a state question, so we're just stating two things that we know. We don't have to link it or explain it or anything like that. But if I'm just thinking back to the other part of the question we've just looked at, then it would be good if it was something that we could say would reduce the number of fish being caught in total, or something that would reduce the number of young fish or breeding age fish being caught, for example. So enough was being left to breed. So these are the two that I'm going for that reason, because I've had that kind of hint from earlier in the question, but there's others in the mark scheme that you can obviously use as well. So to reduce the number of being caught, we can use fishing quotas, and to make sure that only kind of older fish are caught and the younger ones who are kind of haven't yet bred can be left behind is about increasing the mesh size. So increasing the hole size in nets and introducing fishing quotas are my two quoted points. you've obviously got more in the mark scheme you can use, and any of those will get you 2 marks. In 2004, there was a large earthquake and tsunami in the Indian Ocean where the yellowfin tuna is caught. Using the graph and your own knowledge, suggest how climate change could have an impact on the future security of fish stocks. So from the graph, we go to 2004, what happened? So that catastrophic event, that natural disaster, clearly massively decreased the fish stocks, because we've got a massive decrease in the total number of fish that were caught and a massive decrease in the spawning age fish, but also we've got a lack of them being able to recover. So they still haven't got anywhere near back up to those levels since then. So the big that's our using the graph bit. What bit of knowledge can we get from the graph then? That this big natural disaster event, big stormy churny sea event with the tsunami, massively impacted those fish stocks. Now we need to think about our own knowledge here, because it is a suggest question. So our own knowledge here about climate change. So that means more extreme weather or very extreme sort of temperature changes, and how that's going to affect the future security. So these extreme weather changes and potentially, or temperature changes, whichever one you want to go for, could cause these large decreases in fish stocks, because they could cause fish to become extinct, or habitats to change, or they could migrate and move away from the Indian Ocean. There's lots of explanations we can give here that are linked to climate change, and it could cause these large decreases that then may take longer to recover. And that could mean, in terms of food security, because this is really a food security question, there's not enough fish then to supply our growing population of people. We need more food to feed more people. And if we have climate change causing big decreases in fish stocks, then that's a problem. Okay? So this is quite a hard question, because you've got to sort of use the graph to get the clue about a big decrease and a lack of recovery. So that's our kind of graph point. Then we've got to use our own knowledge about climate change. So think about what impacts climate change could have on fish stocks, or could cause fish numbers to decrease. So things like extreme weather, temperature changes, so storms and things like that, or big changes in ocean temperatures or ocean pH or things like that. And then we've got to link that to food security for the third mark. So what do we understand about food security? So having a regular, kind of safe, stable supply, enough to feed the people that we need to feed in an area. And so having an impact of climate change on that could obviously reduce that security. So a large decrease in fish numbers caught, and these numbers were slow to return to pre-2004 numbers. So that's the impact from the graph. Climate change could cause more extreme weather events, which could cause similar decrease in fish populations. They may not be a large, stable catch to supply a growing population. You've got slightly different wording in the mark scheme as well. There's other sort of things you can say to get marks, but this is the general idea. We have the idea of something from the graph that you can get, this idea of big changes, big dramatic events could be causing decreases in populations. The link of the idea of something that you can link to climate change that could affect the populations, and then this link to this idea of food security. In some places, tuna are caught and farmed in captivity inside large pens in the sea. Explain why these fish grow faster than the tuna that are free to swim in the ocean. So it would like reasons why being raised in a pen in the sea or being farmed increases growth compared to being in the ocean. So this is a bit referring back to the energy transfers. So this idea that if you control farming in a certain way by preventing the movement of your organisms too much, they're going to need to do less respiration for the energy to move around, or in case of farming, it would be kind of maintain their body temperature or things like that. So because they're going to not move as much because they're in pens, but also they don't need to go and hunt food because they're going to be given lots of food, that food is obviously going to help them grow faster as well, because they're going to be given plenty of it, lots of it, and be high nutrient food. And also that means there's less respiration required there. So therefore more energy or biomass, whichever you're going to go for here, can then be used for growth instead. So instead of wasting that energy on movement to go and hunt food or because if they didn't catch enough food, etc., in the wild, then they wouldn't grow as quickly. Whereas if we're controlling that environment, then we're making sure that they're not wasting energy, and more of that energy can then go into their growth, their biomass. The fish in pens cannot swim around as much, and they do not need to hunt prey as food is provided. This means less energy from the food is used by the fish, for movement in this case, so more energy can be used for growth. So I've talked about energy here, but you can talk about biomass if you want to. So more biomass from the food is converted to biomass in the fish. That's fine. You've got sort of slightly different wording in the marketing that you can use, but you've got this 4 points, this idea of like they're not moving around, they're being given loads of food, they're not wasting energy moving or catching prey or doing something else through respiration, and so therefore more of that energy or more of their food can then be transferred into their biomass. Give one advantage and one disadvantage for this method as a sustainable alternative to ocean fishing. So one advantage, so why is it better than catching fish from the ocean? And what is an issue that there might be with this type of farming? The first question kind of gave you a clue. You can produce larger fish, or you can get a larger yield. You can reliably have larger fish to kind of farm through farming than you would if you were just catching. You'd get a different range of size of fishes. So that's one thing. There's other advantages on the list, but you just got to think about what things can you control in this environment that can mean that you won't have the uncertainties that you might have if you were just catching fish out of the sea at random with less control over their conditions. And then the disadvantages are all about kind of welfare or ethical or moral issues with this type of farming. So people may be concerned about animal welfare. So some people might not want to buy tuna that has been farmed as opposed to sustainably caught from the ocean tuna.
Well done for making it to the end of this paper, lovelies. I'm really, really proud of all of your hard work.